Hello, everyone, and welcome. Hannah and I are really thrilled to kick off the 2026 Microbial Insights monthly webinar series with all of you. First of all, I want to thank you for joining us and for the level of interest that you always express each year in the MI webinar series. We're very grateful for all the attendees and the interactions that you give, and we're really excited for the level of interest for this month. As you all know, in signing up, we're starting the year with a topic. And for this one, for me personally, it's truly been decades in the making, and I just can't wait to hear your feedback at the end of this presentation. The title is Improving the Impact of Molecular Biology Data for Groundwater Remediation. And we couldn't ask for a better speaker to launch this series than the one that we have today. I'm sure you're all familiar with Dr. Giovanni Pilloni. Dr. Pilloni is the U.S. Superfund Project Manager at ExxonMobil Environmental and Property Solutions, and he's really a leader in environmental microbiology and bioremediation. He spent more than a decade exploring microbiomes across natural and engineered environments, and his work includes over 30 publications on everything from microbial influenced corrosion to biofuels to cutting edge bioremediation strategies like we're going to be able to talk about today. So I'm really excited to have him as part of this series. Before I get started, I want to note a couple of quick reminders. First, this webinar is being recorded, so it's going to be on the resource section of the Microbial Insights website in the next couple of days. We're also going to send out an email with the recording link, so each of you will get it, and you'll automatically get a certificate of attendance for everyone that attends the webinar today. So no need to reach out, that'll be automatic in your email that will come to you. As you are listening to Dr. Pilloni today, can you also check out your handouts section? There's 2 handouts that we've added, a paper that is on this topic, as well as some information on the Quantitative Petro that we're going to be talking about. And then as you're listening to the presentation, when you have questions that you would like to ask, go ahead and enter those into the question and answer panel that's showing there on your sidebar. And if you'll type them in, we'll get to those at the very end. We're also going to add a link or 2 into the chat as we're going through the presentation, so be able to watch your chat box as well. At the end, we'll get to just as many of the questions as we can. If we can't get through anything, everything that we submit, we'll send out a summary email at the end. So without any further ado, I'll go ahead and turn it over to you, Giovanni.
Thank you, Dora, and hello everyone. I'm not even attempting to say good morning or good afternoon. I know Microbial Insights has a customer all over the world, so thank you for joining, all of you. And thanks Microbial Insights for the invite to present this work. Just allow me 1 minute to acknowledge my co-authors. Here in the first slide, you can see Ben Oyston and Trent Key are my colleagues at ExxonMobil. And in particular, Trent Key is responsible in a good way for bringing me into this project. Of course, also Sam and Dora from Microbial Insights, they were really the ones catalyzing, if you want, this work and providing most of the data that we used in this work. And finally, but not just in terms of importance, quite the opposite, John Wilson. I knew John for his work before doing this project, but then really working with him on this was a very important experience, and I've learned a lot from him. But now let's go into the topic, and this is the outline of my presentation. I promise I will spell out all these acronyms as we move forward, but we'll start with the general introduction and in particular focusing on M&A and MBT. Then I will describe the scope of the study, showing how we develop this new estimate for a rate constant based on qPCR data, and how that compare to classic field-based rate constant. Then I will describe you two specific case studies for two fields in Australia and Germany. And finally, more validation, some conclusion and outlook. So in terms of introduction, as I promised, I was going to spell the acronyms. BLUF is a slide quite often used in the corporate world, which stands for bottom line up front. And this is basically just to say that for petroleum and chlorinated hydrocarbons, M&A or monitoring natural attenuation is the most effective remediation method. And this method, especially for petroleum and chlorinated hydrocarbon, is largely driven by biodegradation. And to assess biodegradation, we use MBT or molecular biology tool, and MBT are the only direct line of evidence that biodegradation is occurring. Because with MBT, we detect specific microbes that are involved in hydrocarbon breakdown. What we did in this work was try to develop a new model to estimate rate constant of petroleum hydrocarbon degradation starting from MBT data. And so with this work, we are basically providing a new tool for project manager to improve the forecast of their basically site management. Many of you are probably familiar with what happened in PHC or petroleum hydrocarbon impacted sites, but basically when contamination occur in groundwater, the groundwater flow transport the contaminants along the gradient of the groundwater flow, and many biotic and abiotic process occur at the same time. As you can see in this slide, we can have volatilization, dissolution, and sorption. Especially in the product phase, we can have also some methane production. And then in the groundwater plume, we have largely attenuation processes driven by microbes that are more often than not anaerobic process, because good electroacceptor like oxygen, but actually also nitrate, are consumed pretty quickly, and then sulfate, iron, and manganese are left as anaerobic electron acceptor for microbial degradation. And so, as you can see from this schematic picture, there are many different processes occurring at the same time. And because of that, we use multiple line of evidence for assess M&A at contaminated site. So I will go into the details of this site later in the presentation, but really this is just to give you an idea of this site in Victoria, Australia. Here there is like the contamination plume, and the orange star is where the contamination started. And then if you see the groundwater flow, basically as saw before, move the plume in the direction of the flow. So the multiple line of evidence, as described in this EPA directive that is referenced here, include basically 3 line of evidences. And so the first one is the primary or contaminant degradation. So of course, we need to make sure that contaminant decrease, and the trend of decreasing either through time or space is supported by appropriate sampling. And for this site, you can see that from the higher concentration close to the source, we can see decrease of the BTEX along the various sampling web. Secondary line of evidence is more hydrogeological or geochemistry based, and is an indirect proof of natural attenuation, and also the rates can be used to show the reduction of contaminant. And so similarly to this, to the previous chart, for the same site, you can see some of the most common anaerobic electron acceptor maybe being fully depleted or almost fully depleted in the area where the dissolved room has also lower concentration, and then having higher concentration further down the down gradient. And finally, the tertiary line of evidence is the MBT, which is the subject of today's talk. And again, molecular data from either the field or from microcosms in the lab to demonstrate that the microbes are there and they are performing their work. And again, for this slide, a few examples on benzyl succinate and benzene or benzoyl-CoA degradation genes, showing a general presence across the board. Now, the fact that the MBT are defined as a tertiary line of evidence sometimes relegate this technique to basically a minor component of site evaluation. And of course, we need to evaluate the contaminant degradation. Of course, we can use geochemistry data, but we have to remember that when we do MBT, we use largely qPCR, and more often than not, I see these data being used not in a quantitative way, right? Although the qPCR, the Q in qPCR stands for quantitative. So the question in this work was like, can we use this data more in a quantitative way to have to estimate actually rates of biodegradation at a specific site? And of course, this could be extremely helpful, as you can imagine, in forecast and prediction of site management. So, as mentioned before, for aquifers, we are talking mainly about anaerobic conditions, so in the absence of oxygen, for the most relevant contaminants. And so we choose to start from BTEX, but we choose to start from toluene, especially because the gene for the degradation of toluene, bssA or benzyl succinate synthetase subunit A, are the best characterized among all the other BTEX. And so what was the scope of this study? So first of all, let's step back a little and try to understand what field-based rate constant are and what was the benchmark that we used for our study. So in this study from 2002 from Newell et al., basically there was the not the first, but one of the application of rate constant for M&A. And it's a document with really lots of applicability, but in short, the rate constant are calculated as the slope of concentration versus time or concentration versus distance curves. And so in this work, what we did was mainly use concentration versus distance, but then since we know the groundwater velocity, we can also calculate rate constants with time by converting distance into time. And so to show you an example again for the Victoria site in Australia, these are the toluene concentration on the y-axis in milligram per liter, and on the x-axis there is the distance along the flow path. And so you can see the regression, and the as mentioned before, the slope of the regression give us the rate constant with time, which is 0.02 per meter. And then we also know that the groundwater velocity is about 166 meters per day. And we can convert, using these two, we can convert into rate constant with time at about 3.5 per year. Now, for the benchmark, we actually use the publication from Suarez Rifai from 1999, and they have, like, in this publication, many different rate constants for chlorinated and petroleum hydrocarbon and so on. We selected specifically only 43 first-order rate constant for anaerobic degradation of toluene only from field study as the benchmark. Okay, so from that publication, those are the data that we extract as our benchmark. And then you will see in all the slides moving forward a chart similar to this with rate constant per year on the y-axis. And basically from those field studies, we calculated the 25%, the median, and the 75%. So we use this because every other calculation that we make for our study, it's a good calculation if it fits within this benchmark. And so, for example, if we take now the rate constant of 3.5 per year that we calculate on the left side, and we add to this slide, you can, to this chart on the right, you can see that it fits perfectly within the benchmark. Then we develop a way of calculating rate constant for qPCR data. This is a modified version of the Monod equation for biodegradation. And on top, of course, of mass and time for toluene and volume and so on, you can see what you would expect to add for converting MBT data into rate constant. And in particular, I'm talking about the maximum instantaneous growth rate, the growth yield, and the active biomass. And all these data came from the DGGT culture from University of Toronto, so big acknowledgement here also to Elizabeth Edwards and Cartney Toth for providing this information even ahead of their own publication. So truly acknowledged. And so now let's see when we take the qPCR data and use this equation to calculate rate constant, what happens. So for case studies, as mentioned, the first case study is to this site in Victoria, where we sampled groundwater, and the wells with the blue square are the one that were sampled for bssA gene, and the other with the orange dots are the one used to estimate the rate constant. And you can see that there is an overlap on two wells, and then basically we can cover most of the site. Again, as mentioned before, the orange star is where the spill started, and the shape of the plume is roughly the one in the dark spot. Now, you saw this before, 0.021 rate constant with distance, and these give us 3.5 per year rate constant from field data, so not from qPCR. Now, if we use the qPCR data to calculate the rate constant, you can see these orange dots are the rate constant per year, and here I'm plotting against the bssA concentration, right? And you can see that it's really not that great, right? So in this example, actually, we have a huge distance between the average, the mean of the rate constant calculated from MBT data and the true rate constant calculated from the site, the toluene concentration. And so this big distance was kind of discouraging in the beginning, because it seems that our approach was not accurate enough to calculate the field constant, the rate constant from MBT data. However, the second case was from a different site. So now, similar situation. This site is in Düsseldorf in Germany. Here is where the orange star is where the release occur, and the groundwater flow showed the in this direction show how the plume developed. Now, this site, it was the subject of my PhD thesis, and along with me, another probably 7, 8 PhD students and postdoc did work on this site. So if you're interested, there is a ton of information about this site. Now, my project was specifically about this ML 19202, the one with the asterisk, because this was the location of a multi-level well with the 2 centimeter resolution, where we were characterizing a vertical distribution of contaminants and microbes. However, we had also some sporadic sampling along the gradient, and so we could basically repeat what we did for the Victoria site for this site. The slope based on the concentration is not too different than the previous one. However, with the higher groundwater velocity, we have a rate constant, which is almost double than the Victoria site. And now, if we try to convert this soil MBT into rate constant, now you can see that the difference is much smaller. And actually, the mean of the MBT calculated rate constant is even higher than the field rate constant. But regardless of that, the two values, the two average, are really close in this case. So what is the difference? What's going on here, and why it didn't work this well for the Victoria site? Well, the difference is that the Düsseldorf site was based on soil samples, and if you are at this webinar, probably I don't have to explain you this, but most microbes like to stay attached to surface, and even groundwater soil are an excellent surface for attachment for microbes. And so this is the classic cycle of biofilm. But for work that we did in Germany, but also general knowledge from the literature, we know that microbes in groundwater are present in soil between 10 and 100 times more than free floating in groundwater. And so that was the reason why for my PhD, most of the molecular analysis were done actually from solid sample. And as you are probably aware, this is not uncommon also for our body. This is a nice infographic from Frontiers, and you can see how microbes are ready to attack basically every natural or implanted material in our body with biofilm. And it's also not uncommon in our industry, my industry, the oil and gas. And this is an example from work in microbial influence corrosion that we did Exxon. And you know, if you are in the field of microbial corrosion, you know how important it is to grow microbes on metal coupons. And in this case, we are comparing carbon steel with microbes versus without microbes. And the biofilm is evident and is also causing higher corrosion as compared to the control. And again, if you are in the field of microbial corrosion, you're probably aware that some of this work is also used by Microbial Insights for characterizing MIC in the field. So with that, we also analyze even more samples, and again, Microbial Insight here was basically providing lots of these data points, 183 data points, which is about 60 sites. And again, we also add another soil samples. Unfortunately, there are not too many soil studies for obvious reason, but we were able to add also a bench study from Sun et al, where again we could have qPCR data from solids. And if you then calculate the average, the mean of all the soils and all the water, again, the picture is not too dissimilar from what we saw before. Basically, all the soil sample average appear very close and for sure within the benchmark, while all the water samples are down here. And so what we did was also define acclimation for microbes, as the microbes in the water sample that are capable of basically performing toluene degradation. So we establish acclimation at 0.03 per year rate constant, and this is because it's a 10 times lower than the rough rule of thumb that is used for basically assessing rate constant in the field. And so when we define the acclimation, then 25% of the Microbial Insight database is above acclimation. And why it is important to define acclimation? Because, of course, we understand that sampling soil is not always possible, is not always convenient. So, of course, our recommendation is if you can sample soil and if you can get MBT derived rate constant from soil, that will be more accurate. However, water samples are more common, and if you don't have soil sample, you can still calculate rate constant with water sample, and if your rate constant are above acclimation without including any well with residual NAPOL, then in a sense you are justified to use benchmark rate constant. And you can see actually an application here on the right side. These are now data point that I didn't have at the time we wrote the manuscript, but then I obtained this data from a colleague, and you can see that site number 1 is below acclimation, so we don't really consider it. And site number 2 and site number 3 are above acclimation. And when then you calculate the field-based rate constant for site number 2 and site number 3, you can see that basically you are well justified in using the benchmark because these two rate constant for these 2 sites are within the benchmark. And in a sense, what I find also intriguing is that they seem to have some ratio, they seem to be proportional to the MBT calculated rate constant. Which in turn is extremely important because, for example, for site number 2, we could use a 7 times higher, and for site number 3, almost a 50 times higher rate constant than the 0.3 that is normally used as a general rule of thumb. And for site number 2, where the remedy is actually MNA, we can even forecast toluene to be undetectable in 5 years based on the rate constant calculated by using the benchmark. In terms of path forward, of course, we know and we are aware that toluene is often not the main contaminant and the main driver for sites, but it was a good place to start. So we need to go for the difficult one, especially the toluene, and we are starting some work to move in that direction. Also, it was working with John Wilson was a good reminder that the field that estimate rate constant do not require MBT data, and still we probably don't use as much as we should. And also that there are similar models for chlorinated hydrocarbons that can be used. Of course, now, as you can see here, I'm finding internally more user cases, but I also encourage everyone in the call who has interest to apply these. And to apply these, of course, you can refer to the paper. This has been published a few months ago, and it's also in the resources in the attachment of this webinar. But also, of course, I understand that not everyone wants to go through the equation and through those calculations. So Microbial Insights has been so kind to put a calculator. Basically, you can download on your computer the same calculator that we use, or what, a similar calculator to what we use for our manuscript, and you calculate your own rate constant for bssA and toluene concentration. And so with that, I hope you have questions. Thank you for your attention.
All right, thank you so much, Giovanni, and thank you, everyone. If you can go ahead, we do have a few questions that have been entered into the GoToWebinar panel. If you have others, please go ahead and enter it. Also, if you note in the chat box, Hannah has added the link for the calculator that Giovanni just showed you so that you can go directly and check out that page. So one of the questions we have from a couple of people, Giovanni, is can you talk a little bit about what you mean when you're talking about acclimation and the importance of that first, what is acclimation and how did you come up with that? And then second, what is the importance of the 3% rate constant?
Right, so the acclimation itself is basically in 4 wells where we don't have any residual NAPL, where the dissolved contaminant meet electron acceptors and the condition are adequate. It's basically those wells we define acclimated for biodegradation. So it means that basically the microbes in that well are capable of converting the contaminants and basically degrading the contaminants. Now, the 0.03 is, I would say, it is not a magic number. I mean, there is nothing written in stone about this number. It's simply 1/10 of what is currently used in the field. So in the field, when we want to calculate rate constant for toluene degradation, well, it's actually a rule of thumb pretty much. So, and 0.3 is used for basically as rate constant to calculate toluene biodegradation. Now, since all of these calculations show that if you use MBT, so bssA's concentration, to calculate a rate constant from water sample, you are most likely underestimating because your microbes are not really in the water. They are more often than not attached to the soil. Then we decided to use that 0.3 divided by 10 to account for the underestimate. And so by doing that, we include way more samples from the water phase that we would if we were going to use the classic 0.3. But again, it's not set in stone. As you can see here, there might be also some future application in which we can refine this acclimation value based on additional observation. And yeah, the fact that 25% of the database is above acclimation, it is actually pretty good, because if you consider that above benchmark we have only like what 4 data points from the water, that means that actually by using this acclimation value we can include way more data point from water samples, and if our samples are above acclimation, we can use the benchmark value from the literature.
All right, thank you, Giovanni. There are a lot of people that are saying that this was a great presentation and much needed work. One of the questions that they're asking is something that you and I talked about even right before this call is, you know, we're really focused here on anaerobic degradation of toluene with the bssA gene. A lot of people know that benzene's a regulatory driver at their sites. And so the question is, you know, how do we expand this to benzene or to other contaminants of concern? As part one, and then also part two, is there a way to, even for toluene, to look at more aerobic degradation markers rather than just focusing on anaerobic? So maybe if you can talk about what we're thinking there, and then the complexity, I think that would be helpful.
Absolutely, and these are excellent questions. And you know, I was at the end of the talk, I was saying that we on purpose choose like toluene and bssA because they are the best characterized, right? Which means that we don't have genes for benzene degradation that can capture all of the diversity of benzene degraders anaerobically and strictly speaking for benzene. And you know, it might be maybe halfway in between for ethylbenzene and silenes, but really, and of course, I would say that for aerobic, when there is an aerobic condition, this could be applied for aerobic as well. Now, Dora mentioned that we discussed this in the past. It's true. I mean, really, this is like a first step, right? So it's not the final solution. It's a good one. I mean, for toluene, I'm confident it works, but it's a first step. So the dream, if you want, would be to have like a multi-parameter kind of modeling. You see the calculator that is now on Microbial Insights website. Ideally, you would copy and paste your entire list of contaminants or your entire list of degradation genes, and you will have like consolidated output with rate constant predictions and so on. For some of these genes, we are not there yet. We need more research, and you know, there are hopes. There is Elizabeth Edwards and Courtney Toth. They are still working on this, and I'm sure they will find more targets. But that's really the issue here. The reason why we cannot do benzene now is that we are on top of the underestimation of using water sample. For benzene, we are most likely underestimating also the microbes, because we have the assay that Microbial Insights use, which is really good for a specific group of microbes doing benzene degradation, but we are probably not including some of the other microbes.
Yeah, I mean, to add on to that, we know one pathway, right, for benzene anaerobic degradation of the carboxylation step, but we our researchers are still working on and have spent, some of them, the majority of their career working on trying to understand that hydroxylation step and other ways that anaerobically benzene can biodegrade. So until we get those biomarkers, we can look at parts of the degradation pathway, but it's we're kind of limited by not knowing the direct pathway to be able to do a good job of investigating that. The other thing too, Giovanni, and maybe you can expand on this as I start, one of the questions is, you know, do we have a reason that we focused on anaerobic biomarker for toluene? And I think, you know, from my perspective, a lot of the sites are pretty anaerobic, especially in the source area of the plume, because you get so much depletion of electron acceptors that are available once you have a release. And there are a lot of aerobic pathways, so it was a little more complicated to have enough samples that had more aerobic conditions for us to be able to look at those biomarkers, plus to be able to look at the diversity of biomarkers. So that's, you know, kind of some of my thoughts on that, but wanted to see if you can add value to what I said there.
You know, absolutely. Again, my major experience was with this site in Germany, and as I was mentioning, through this ML 192202, while we were analyzing also the vertical profile. So, agree with you. Oxygen is very limited in most sites. In this particular site, we were able to find in the very first centimeter of the plume still some influence of oxygen vertically. But then as we were going down, sulfate and iron became the major electron acceptor. So yeah, to confirm what you said, today in the site that I manage as an environmental portfolio manager, I almost never see petroleum hydrocarbon plumes with oxygen available as electron acceptor. And you know, that's true even for some very shallow aquifers, and that's exactly the reason you were mentioning, Dora. Oxygen is quickly depleted, and it doesn't reach the depth, quite often doesn't reach the depth where the contaminant is. So for the attendees who have oxygen in their sites, I would say calculating rate constant, even using the Todd genes or whatever, should be feasible because those pathways are pretty well characterized. But of course, our focus was mainly on the anaerobic component of groundwater, contaminated groundwater.
And if there are people out there that have aerobic sites that they're interested in, if you want to reach out to me, we can talk about that. We do have a lot of biomarkers that we could use, and we can try to pull some database information and kind of help you with understanding what that means. So reach out if there's specific examples of that. And Giovanni, one of the questions people have quite a bit, there's several variations of this question, is really understanding why you mentioned that the presence of residual NAPL can have an effect on using this calculator.
Yes, so again, it goes back to the acclimation discussion. If a particular well is within the NAPL, within the product, it's you can still have degradation, but it's probably more methanogenesis than anything else. So really, it's not a bssA or any other anaerobic gene, well, except methanogenesis, driven process, and so that's why we were trying to exclude those wells from the calculation, if that makes sense.
It does. And I think, to add to that, natural source zone depletion is the process that Giovanni's mentioning there, which is highly driven by methanogenic conditions and surface interaction. We do have a quant array that can evaluate that. So if there's interest, you can reach out and look at that to help you evaluate NAPL situations. The fringe area of that where you get degradation from NSCD, you will get some production of toluene and lighter compounds that then we'll see bssA and these other biomarkers come into play. So maybe it's on the edges of where that NAPL is, but for this, for this particular calculator and doing this, to be sure we were only looking at degradation that was related to this particular gene in this situation. We really kind of excluded a lot of that data just to make sure we were very focused in this study. But I think, NSCD is certainly something you should think about if you have NAPL that's present in those. And I don't know if you have any other experience on that, Giovanni, that you want to add to.
No, absolutely. Not much of a direct experience, but indirectly working with other colleagues. I've seen characterization of NSCD also. And yeah, as you said, we also use on top, of course, of the measurements of methane, but also some of the quant array assay. And this was, in fact, the case specifically for this site in Australia, where both NSCD and natural attenuation were occurring at the same time. However, as Dora mentioned, specifically for this work and specifically for looking at bssA gene, we decide to exclude methanogenic condition from these calculations.
One of the questions, Giovanni, that was submitted, and it's something I haven't really thought through, but maybe you'd have some ideas on, since we don't have a lot of soil data to be able to help us in these situations, and coring can be so expensive that it's not common in site assessment. If people used biotraps as a solid substitute, I know they're not going to give you the same longevity as soil, but do you think that would be valuable? And maybe we should take a look at the biotrap data compared as an intermediate between soil and water.
It's an interesting question. I've never used biotraps, although I've been always intrigued. Since the time of my postdoc, I was always wanted to use. But in general, yes. It's sort of simulated soil at that point, right? So you're offering to microbes a surface for attachment, and then try to understand if it's similar to soil. The question is understanding then the rates of microbial attachment and deattachment, because again, biofilms are not something that it happens and then it stays always the same. Based on nutrients, based also on quorum sensing and many different processes, a biofilm can form and also being released, right, at any point. So we need to understand how that works. And to be honest, when I was looking at biofilm for corrosion, we don't know everything. The medical microbiology basically is where the biofilm are best characterized, and we know signaling molecule that can initiate or terminate biofilm. Actually, even drugs are target against those. But then, as often happens when you move to the environmental side of microbiology, some of those enzymes, some of those processes are not as well characterized. And that's why we were doing such fundamental work on corrosion to try to understand how biofilm form. So yeah, long answer to say yes, it's a possibility, but we need to understand first how those dynamics between the water phase and the biotraps and the biotraps and the sediment and the soil works and interact with each other.
I think that would be an interesting future study to do. Another question, Giovanni, can you talk a little bit as a site manager about how you begin to distinguish acclimation phases from times where maybe the compound is more recalcitrant and not degrading as it should?
Interesting. So yeah, this work is pretty recent. Pretty recent is also my position as remediation manager. However, maybe we can go back to the example here from my colleague, right? So here, of course, we also have the site rate constant, right? So the rate constant that is calculated from toluene degradation. However, if for 1 second we think that we don't have those data, what I can see here from the water data and from the MBT on bssA, that actually the green dots seem to be higher, like the red constant seem to go higher as opposed to the orange dots. So of course, in this case, these are partially above acclimation, but even without the acclimation, in this case, I would look at this data and I would come to the conclusion that site number 3 is more, the rate constant are higher as opposed to site number 2. And this is what is then confirmed by the toluene field data and the rate constant based on that. So again, as I mentioned before, acclimation is a convenient check to have right now, but it doesn't mean that in the future we will always use this 0.03 value, right? As I mentioned earlier, it might be that we can refine, and instead of having like one single line, we will have like multiple of this line, and then those threshold will bring us more or less closer to either the 75%, the median, or the 25% percentile. And then, we can have closer to reality rate constant. I also not sure if we will ever reach a point where we have so much data that basically our rate constant can be calculated directly from the bssA concentration without even thinking of acclimation. But really, we're using acclimation as a way to overcome the underestimate of water-based data. But really, even if they're underestimated, you can see that they might still be indicative of a quantitative process happening on site, as it is shown like in this small data set from a colleague.
And this leads into a question that was presented about the first site when you talk about the bssA genes, the field rates versus the MBT rates, you're seeing differences, and you attributed it to differences in attached versus planktonic bacteria. Do you think there are also differences because other genes than bssA might have been part of the biodegradation pathways that were happening?
That's for sure a possibility. I mean, again, we are calculating from toluene and bssA, right? Now, it's either other genes, but it's also possible, and we know that bssA could be responsible for other contaminant degradation. And of course, here nothing as in any site, but also toluene degradation is not happening in a vacuum. It's toluene degradation, and then at the same time that might be silanes and ethyl benzene and so on. And there is even the possibility that the benzene could be methylated and then become a toluene again. So bssA can be used also in that case. So of course, it's not a silver bullet, but in this case, it's equally distributed across all samples, right? So I believe that all the sites represented here are in the same situation. So all of them have BTEX, not only toluene, and then of course bssA is doing the best job it can in detect toluene and possibly other contaminant degradation. And also at the same time, for all of these samples, there might be other genes and other microbes that can add additional noise to this data for sure.
Another thing that I'm seeing in the questions, Giovanni, is people are really excited about having this available, and they really want to see it for benzene. And we know it's a regulatory driver for you guys and something of interest. And so I do want to, give Giovanni a chance to address it a little bit more, but you're talking about the bssA gene in relationship to benzene biodegradation. We certainly know that. We certainly have a lot of data for that gene, and it is something that's on our radar to do. We just haven't got there. I know our group has discussed this a bit, but it is something I think we want to do that as a next step to be able to provide to you guys. But I wanted to see if Giovanni has anything else to add to that, since it is a hot topic of discussion.
Yeah, we actually, Microbial Insights folks tried also to assess other contaminants from the bssA, and we are not putting out there because we want to be sure. So we can try that, but also this in the same time, what I was mentioning before, continuing with the R&D, continuing with the understanding of how benzene degrades, it is important. We are working on that, others are working on that. So hopefully we can have a comprehensive array for all the BTEX and for all the other contaminants. And hopefully that calculator from Microbial Insights at some point is going to be a calculator for all of your contaminants.
Yeah, we hope that we'll see it grow, and using, like as Giovanni mentioned, we did look at not just toluene, but other compounds that can degrade or maybe degrade simultaneously with the toluene at sites. And we certainly have data for the ABC gene for the anaerobic benzene carboxylation that we want to look at. But this is, first step in being able to get something. And from my perspective, after working on this for 25 years, I'm excited to see this first step out there. So I feel like it's a huge stride to be able to make some type of correlation between MBT data and field rates of degradation. But we definitely understand that this needs to go a lot further than what we've done here. And I personally am excited to see what we can do next.
Absolutely.
Just as we wrap up today, I have one more question that was posed out there, and it said, have you thought about using like carbon-14 or carbon-13 labeled substrates to be able to look at rates of degradation? And so I'll give you the MI experience and then maybe pitch it to Giovanni to kind of wrap that up. We do a lot of carbon-13 degradation of toluene, so we have a lot of data that we've done for this over the past 20 years, looking on biotrap samplers, adding carbon-13 toluene and looking at degradation in organisms that are doing it and calculating first-order degradation rates in the field. And I think the good of it is we have data that can give us some indication. The bad is because when you add a 13 carbon compound like that and you make it either in a biotrap or in a bottle, you make it really bioavailable to these organisms. So that rate is a little different than what we truly see in the field, but I do think it's a good start of helping us to understand degradation pathways and what's occurring. I just don't know that it gives us really solid field dates like we're trying to get here from this field data. But I wanted to kind of pitch that out there, Giovanni, to see if you agree with me and what your experience is.
Great point, Dora. I don't have much field experience with the 13C. I used as a substrate for DNA SIP experiments, so stable isotope probing experiment in the past, and I would see value in that back to what I mentioned before in characterizing microbes and understanding the processes. Clearly, if you have a labeled substrate, then the microbes are labeled, the DNA and the proteins are labeled. So that's going to help in characterizing what needs to be characterized. For the field application, Dora, I trust what you're saying, and it makes sense. Of course, now you are enriching. You are a little beyond the natural concentration of that compound, and so it's going to be maybe not 100% indicative of the rates on site.
Well, thank you so much, Giovanni. It was really fun to have this presentation from you and to have this chance to get to chat with you a bit. And I encourage those of you that joined us to reach out. If you have additional questions or thoughts, you can always reach out to Giovanni directly, or you can email us at info@microbe.com. We'll be happy to have further discussions and would love to talk to people that are interested in creating this calculator for other compounds of interest. So definitely looking for other collaborations to continue this work. Before we wrap up, I do want to encourage you, this is the first in our webinar series, that you log on to the MI website to see if there's other webinars of interest. Next month, Dr. Kate Kierzik with Battelle is going to be talking about recovery of rare earth elements with a technology that they've created. I think it's going to be a really interesting talk, so I would encourage you to sign up for that and to look for future webinars each month that are going to come out from MI. But Giovanni, you were spectacular as a kickoff, and I think this was a great topic to start our year. So I am very grateful to you and just want to give you a very, very large thank you from my heart for doing this webinar for us.
Thank you. Thank you for inviting me, Dora, and thank you everyone for attending.
All right. Thank you, everyone. Globally, I see Saudi Arabia and other countries that are on here. So I'm grateful for you guys for participating and hanging in with us. Please check out the recording and reach out with questions. All right. Thank you again, Giovanni. Thank you, everyone.
Thank you. Bye.